Yeah, sure. So my name is Ferdinando Pucci. I'm assistant professor here at OCSU in the Night Cancer Institute. I grew up in Italy, was raised and born, did my PhD in Milan, and in the laboratory of Michele De Palma and Luigi Naldini. They are better known for gene therapy and genetic engineering, and so that's what I learned. I took that expertise with me for a postdoc in Boston at Harvard Medical School in the laboratory of Michael Pitet. And there we had a lot of fun trying to figure out new approaches to investigate these so-called extracellular vesicles in vivo in their native environment. I did a short stint after my postdoc in a biotech. You know, Boston is the world biotech capital, so it was kind of almost impossible to avoid that. And I got into a small startup. I think I was like a third employee, and that was a great experience. It taught me a bunch about translating research for clinical applications, and it also introduced me to the world of T cells. After that, I started applying for faculty positions, and I was lucky to be recruited here at OCSU by Dr. Lisa Cousins and Dr. Flint, the chair of the otolaryngology department. And here I am. So probably, as you know, and most of our audience know, there is a lot of excitement about immunotherapy. It's a breakthrough therapy. We are starting to talk about cure cancer. There is a Nobel Prize, two Nobel Prizes actually, that have been awarded to well-deserved scientists that pioneered this field. And it is not just about the Nobel Prize for their achievements in the so-called checkpoint blockade therapy, but it's also a lot of gene therapy approaches with CAR T cells that enable the high fractions of patients with liquid cancers like lymphomas to be cured. With solid cancers, the situation is still promising, but it's not as good as with liquid cancers. The cure rate for the complete response rates for solid cancers in patients treated with immunotherapy is still, 10–20% range. So there is large margin of improvement. My laboratory investigates, the main theme of the research program is the role of extracellular vesicles in cancer, both in relation to immunotherapy and with early detection. Extracellular vesicles, as you may know, are kind of small particles, membrane-bound particles that pretty much every cell type releases. Tumor cells, although, release much higher numbers of these particles. And this is a very interesting phenomenon because the role that may have in communication between tumor cells and host immune cells, other host normal cells. And this communication is fundamentally different from the traditional communication that we all study in textbooks, for example, in which you have one single signaling element that is released by one cell and sensed by another cell, either locally or distant, in a distant place. Extracellular vesicles carry multiple signals clustered on a very small surface. And so whether these signals are all of the same type or they also integrate different signals, this is something that communication via extracellular vesicles can achieve. And it's really not clear what the consequences of these abilities in communication are, especially in vivo, where these extracellular vesicles are very difficult to study. And this is where my lab is starting to make headway. Basically, we developed models, genetic models, to avoid touching and isolate these vesicles in vitro and then reinfuse them into animal models. And so we just track them how they are in vivo. If you wish, this is kind of, this type of communication mediated by extracellular vesicles is like a Web 2.0. So it's really the next generation of the next level of our understanding in how intercellular communication works. And we are applying these, as I said, in the context of cancer early detection and immune response to cancer, because these extracellular vesicles carry tumor antigens. And as you may know, tumor antigens are very popular now. They are a focus of a lot of research to understand how we can use tumor antigens, tumor neoantigens to better target even solid tumors with immunotherapies. Yeah, I'm really grateful for the V Foundation for funding this research. It's focused on developing therapeutic cancer vaccines. As you may know, cancer vaccines are another hot topic in immunotherapy. The problem is that the response rate is even lower than with the current checkpoint immune therapies. They are really in the single-digit efficacy. There is really, the hard part of delivering the promise of curing cancer via a cancer vaccine is that, vaccines work very well, like, for example, COVID or other diseases that have been also eradicated by vaccines, right? But those worked because they were preventative vaccine. They were administered before the disease could strike to prevent it. Now we're talking about a vaccine that needs to target a disease that is already present. And so the difference is that now there is a tolerance for these diseases or cancer. Our body is used to have these tumor cells growing, which, as you know, takes decades to develop. And to break this tolerance, that is a central point of designing proper cancer vaccines. And we're trying to figure out how extracellular vesicles may play a role in this tolerance induction and how we can revert those processes to break tolerance and have the ability to develop an effective therapeutic cancer vaccine that can work even if the disease is already present. And this is the work that we have been funded to perform by the V Foundation. $1 million question. So I think the direction will be in simplifying the manufacturing. And so having therapies that are less expensive for both the healthcare system and the patients. So, for example, off-the-shelf CAR T cells or off-the-shelf NK cell therapies. The biologics will also play a huge role with the advent and the engineering of therapeutic antibodies and the development of bispecific antibodies and different conformation that can engage the immune cells with the tumor cells. And then there is the area of cancer vaccines. As we progress our understanding of how the immune system works and how we understand this complex biology and how the decision within the immune system are taken, who takes central decisions, who are the foot soldiers that attack the tumor, we will get better to understand what is needed to develop cancer vaccines. And so this will be the further edge of immunotherapy in the future, I believe.